Good evening, viewer. Welcome to our Glory Globe TV. Here is a breaking and most amazing news too for our viewer. A 3D view of the genome. Yes. In this picture, see looking at models of single cell 3D genome structures of human cells, which they were generated using software for molecular dynamics simulation called ChromeFrog, developed in their lab. It issues various data as input, including those obtained by a recently developed method that makes it possible to map how DNA is spatially organized in the cell nucleus. They joined human Technopole last year from the CLIVE laboratory at the Karolinska Institute in Stockholm, where they have been leading their own research groups since 2015. They decided to embrace the adventure, keeping a dual appointment. A combination of private and professional life triggered this choice. The Functional Genomics Research Center at Est seemed the right environment for both them and their Italian husband, who is also their lifelong collaborator. They fell in love with 3D genomics when they were at the Massachusetts Institute of Technology because it's an ideal field for interdisciplinary studies, of which they have a big band. It needs biotechnology, physics and computation. It also requires technology development in microscopy and sequencing. 70 years after the discovery of the structure of DNA, it is time to focus on the entire genome and to understand the function of its spatial arrangement in the entire volume of the nucleus. They want to better understand how the position of genes affects their expression. There is a general understanding that the more they go towards the center of the nucleus, the higher the gene expression is, but it is not a black and white scenario. Indeed, there are also highly expressed genes sitting at the periphery of the nucleus. Disruption of the 3D organization of the genomes can have big effects on our health and is factored in cancer or neurodegenerative disorders. Translocation that result in unusual rearrangement of chromosomes lead to pathologies. But to understand how that happened, we need to understand how they were, and also they are linked to the structure of the surrounding genes. In the lab, they make predictions starting from their sequencing data, and they test their with tools to study chromosomal organization and visualize messenger RNA transcript. The lab has been recently awarded with the ERC consolidator, and this will allow them to go deeper in the analysis at single cell level and trace the 3D genome sequence and structures during cell division. They would like to investigate the role of nuclear RNA in shaping genome radiality, that is the spatial arrangement of genome elements with respect to the cell nucleus, and how in turns genome radiality has an impact on nuclear processes. They are setting up their lab trying to create an environment that is as much international as possible. Something magic happens when people find themselves far away from home and outside their comfort zone. That is also their personal story. Yes, they are, and they are the nucleus is Poland. What they studied? Biotechnology. Then they took their PhD at the Goethe University in Frankfurt and did their postdoc at MIT in Boston. In recording for their lab, they abandoned the philosophy of finding the exact person with the exact specific set of skills for what they need instead. They usually capitalized on people with the right mindset and on their talents. Moreover, they always bet on interdisciplinarity and try to surround themselves with the most diverse backgrounds such as biology, biotechnology, medicine, physics, mathematics, and engineering. Beyond the report, Glory Globe TV, Delhi, India.